Thank you. Before I start, I would like to thank the organizers for inviting me here to Bologna, and also for presenting my person. Today I would like to speak about the AFM applications in medicine and biology, and in fact it will be some sort like a excursion through the work which we are doing in the lab, which is located, unfortunately or fortunately, at the Institute of Nuclear Physics. However, we are not dealing with nuclear physics at all. I mean, we are not doing this. We are working with AFM, trying to get to bring this device to the hospital, and our initial aim was to make the diagnosis, so just to show to medical people that it is possible to diagnose pathologically altered cells also to medical doctors. So we started with some sort of the determination what are the physicochemical properties of normal and cancerous cells. Then we try to understand what is the influence on the environment of cells, because cells, they are not as a single unit which are floating in our body. They are embedded, as Ricardo was saying, in the extracellular matrix, and then we would like to see what is the role of this extracellular matrix in the cancer progression. Of course, cells is not uniform material, it is neither isotropic, neither purely elastic, so that's why we would like also to understand what is the role of their internal structures in the cancer progression. And of course, as I said, the main goal is to find some biomarker of cancer progressions. As we know, already have heard here, AFM have different functionalities. One of the most basic is just making the imaging, and I'm gonna to show only two examples of such a application. However, and then I will move to force spectroscopy, because most of our research we are making force spectroscopy, and the force spectroscopy we are applying to two types of measurements. First, the determination of adhesive in general properties of cell surface, and then to nanomechanics. So starting with topography, topography is less understood by medical world or biological world, because we have other tools which are much better than atomic force microscopy, which are faster, and also they are easier to interpret by these people. So there is optical microscope, there is NMR, there is X-rays imaging. So this is, for medical doctors, it's much better to understand this kind of the images than the AFM. Nevertheless, in 2010, there was a paper showing how surface of red blood cells changing with age. We all know, I mean, at least I know that the red blood cells, they are living for 120 days. And as you can see from this image, it was here. Roughness of red blood cell surface changes with the age of red blood cells. So potentially changes of cell surface could be used as a biomarker of something. Another story is to show or to monitor what's going on with cells which are undergoing certain treatment. Here there is a prostate cancer cells which was treated with vinflunin. Vinflunin is a medical anti-tumor drug which is currently used to cure also prostate cancer. And then you can see nicely organized actin cytoskeleton, which is which is presented here. It is somehow disappearing and reorganizing upon the action of anti-tumor drug. So this is two examples showing what we can really use for how we can use the topography to determine some changes which are occurring in cells. But as I said, I would like to go to force spectroscopy. First, let's start with adhesion measurements, and these adhesion measurements can be divided into two types of measurements. So we can measure the interaction between two molecules. This is called, oh, sorry, it shouldn't be. Yeah. This is called single molecules for spectroscopy, in which we are dealing with two isolated proteins. One is deposited on an AFM cantilever, and the other is deposited on a substrate. And the other technique is just single cell force spectroscopy, in which tipless cantilevers, they are somehow modified with a single cell, and the single cell states force probe, and then using the cells, then we can probe properties or adhesive interactions of the cells with some substrate. Also, there is some sort like an intermediate state, which you can somehow use to measure two types of interactions. So either you studied directly the molecule-molecule interactions on the cell surface, or you can modify it AFM probe with single cells, and then we can measure the interaction between the two cells. However, nevertheless, independently what we are doing, we are measuring the force which is needed to detach something from the substrate, as it is shown in drawing of one of my PhD students. Whatever we will do, we measure the force or work which is needed to detach something from the substrate. So therefore, there is a need to analyze the data in a specific way. In case of single molecule interactions, it's relatively easy. This means that you are trying, sorry for this, but somehow I'm stupid with this pointer. We are looking for this kind of shape of the force curves, which is recorded during withdrawing of the cantilever from the surface. So this is the part of the curve which is in contact with the surface. Then we try to unbind our cantilever from the surface. If there is any adhesive interaction, deflection going to the opposite direction, at certain moment our force is large enough to detach cantilever from the surface, and then deflections comes to the zero position, to the base level, where deflections are negligible. From such a kind of image, we can determine the unbinding force, we can determine the stiffness of our molecular complex, but this is not the proof that you are dealing with the specific interactions, with the interactions which are occurring between two single molecules. In order to do that, we have to remeasure again our system in so-called inhibition conditions. These inhibition conditions, the idea is very simple. So we have some ligands which are attached to the AFM tip. We are adding the same ligands as in a free state as free molecules to the solution, and then we are waiting for a moment. Free molecules, they bind to receptors which are present on the surface, and then the interaction should be inhibited. This is realized or visualized in that case. Then, sorry. In that case, on such a plot in which we are plotting the number of unbinding events versus force, and if we have our normal conditions in which we can observe some interactions, then if we, thank you, and then if we provide inhibition condition, then the number of observed events should decrease. In such a case, there is, this plot, there are comparison of three types of the inhibition ways. So first one is just to add to the solution monoclonal antibodies, which bind to the receptors, and they will block them. And as you can see, this is this dark blue histogram. It really, the number of events decreases close to zero. The same efficiency is observed for conditions where 10 millimora EDTA is added. Slightly different results you can get if divalent ions will be exchanged into monovalent ions, but this case is really dependent on the type of the molecules which is studied, because some molecules, they require calcium ions to bind, and some molecules not. In case of the measurements conducted on cell membrane, the situation is more different because not all conditions can be applied to living cells because you will kill them. In such a case, the most common approach is just to use 10 millimolar EDTA, and then we observe the decrease of unbinding events. However, as you can see from this plot, it really depends on the cell type. This is a transitional cell carcinoma, and as you can see, adding 10 millimolar EDTA for these red columns doesn't change much. So in that case, we can say that there was no specific interactions. In the case when we see dramatic drop, like for this bladder carcinoma grade 2, then we can see that what we measured, it mostly was our specific interaction we would like to study. In the case of cell single-surface spectroscopy is somehow different because, cell surfaces, on the cell surface we have huge number of different types of molecules, and there is a question how to select this particular one we are going to study. So first of all, this is a plot showing melanoma cells which was attached to the cantilever without tip. We are getting more complicated force curve, but nevertheless you can get the information about the force which is needed to unbind the cell from the substrate. We can calculate the work of an adhesion, of energy of adhesion, depending on the school. And also you can determine what is also popular, the distance, which tells you what is the size of the cell membrane which was protruded from these cells. To verify this data, the common way is just to measure carry measurements on two types of surfaces on which we are expecting specific interactions to be dominating, and on that in which we are non-specific interactions will be more common. This is the results of such a work, and then BSA albumin is the most common protein which is used to show the specificity of the interaction. The case that it is the work of adhesion drops here is not a very specific one. It could also be that the work of adhesion will be higher from our desired measured interactions. In that case, for this HCV29, we've measured the adhesion force and the work of adhesion between the human bladder cells, non-malignant one, two types proteins of extracellular matrix, to fibronectin and vitronectin. As I said, on cell surface we have different types of receptors. Here there are two of them which we studied further. They belong to the family of integrins, which are, everybody knew more or less. And there's also another family which is called syndecans. And then when we compare the adhesion, then we can see that as it was shown in 2010 in the work of Hozumi, then both receptors are needed to have cells which are nicely spread out. I have to say that if we have cell cultures in cells, they are rounded, so this means that the conditions are not very good for them, so they will not spread out. Cells which are in good conditions, they should be very nicely spread on the given surface, and this gives the information that the culture was performed in a proper way. We observed the same for our bladder cancer cells. This is the results showing that surface area is really dependent on the type of the receptors. So have fibronectin, glass with fibronectin, glass, and glass with syndecan, with monoclonal antibody against syndecans. And then you can see some changes in the surface properties. However, this is also the situation that we can enhance surface with certain types of the molecules, but still we have a lot of information on cell surface. So we cannot remove receptors from cell membrane. So therefore, we start to make measurements using the isolated system. So first, we quantify the interaction of syndecans using monoclonal antibodies. Why monoclonal? Because monoclonal antibodies, they interact specifically with the particular type of the receptors. Then this is our results. Using two theoretical models, Bell-Evans or Dutko-Hammer-Szabo. I mean, I am just showing the results for Bell-Evans models. Then we see that the unbinding of syndecan 1 proceeds through two energy barriers, because there are two linear regions observed in our dynamic force spectra, and the interaction between, for the syndecan 4, this is the fourth member of the syndecan family, and the interactions reveal only one linear region, so it means that there is only one unbinding, one energy barrier in the energy landscape. This could be attributed somehow to the structure of syndecans. So most of the syndecans, I mean, the syndecan 1 is the longest one with very branch chain, while syndecan 4 is shorter one, and also the length of this chondroitin or heparin chains is smaller. In addition, when we look in our bladder cells, we see that the amount of syndecan 4 is much higher in cancerous cells. Cancer cells always I'm putting in some reddish color. And in normal non-malignant cells, which resembles normal bladder bladder cells, there is the amount of both syndecans is very low. So we see that cancer progression is not changing only, the amount, which is common way to determine some expression of some molecules and saying if we have something higher, then we have cancer. If we have the lower level of something, then we have no cancer. But also it changes the unbinding properties. Then I'm coming back to my verification experiment, because you can get also some other information that syndecans, which were believed to be, they are proteoglycan receptors, and there was no information whether they require calcium divalent ions for binding or not. And our experiment with inhibition experiment with different types of inhibition buffers show that syndecans are calcium-dependent family. So they required calcium to bind properly. If they would not require the calcium, then the replacing divalent ions into monovalent ions would not change the shape of the histogram. So these two, both histograms red and this green, blue light would be the same. And then we compare also next interaction in which we try to compare the unbinding force which is needed to unbind integrin from extracellular matrix and syndecan from the extracellular matrix. And it appears from this plot that also this is a different way of unbinding. And then we plot some sort like an energy landscape, which show us that in fact we have that the binding of syndecan to extracellular matrix is more stable. The syndecans, the complex is very soft. Integrins complex, which is done with integrins, is more stiffer. So when we look at the sequence of unbinding, then first this is an integrin who unbinds, and then syndecans. As I said, also, we studied this extracellular matrix because we would like to understand what is the role of this environment of the cells in cancer progression. And indeed, there are some literature information showing that if we have extracellular matrix, some cancerous cells first, they have to do something with basal membrane. Basal membrane, this is a membrane which is composed of the laminin. And in general, epithelial cells, they are not going through This is like in our brain, we have a blood-brain barrier, which is also contained basal membrane. So our cells which are coming from immunological system, they are not going to the brain area. Within the brain, there is a specialized immunological cells. If everything is working properly, basal membrane cannot be open. So cancer progression just do something with the basal membrane, and at a certain moment it breaks. I mean, do something means that they are changing frequently laminin into collagen. In most cancers, we see some abundance or high expression, more amount of collagen fibers. And in addition, these collagen fibers, they are stiffening the extracellular matrix, and cancer cells, they can walk using this collagen matrix and go further, of course, to make some metastasis in distant place. Also, when we, in the simpler image, when we look, this is the extracellular matrix, which is dense. Cancer cells first, it has somehow go through the primary tumor site to our blood or lymph circulation system. When we think about the physics, so at this place, single cells feels compression, tension, some shear forces. But if we are going to our, our veins, then there is no compression, or compression is negligible, then we have more shear forces. And then again, when it starts to colonize other distant place, again there is a change from the type of the mechanical forces single cells is sensing to another type. So that's why it's good to see something, to know what is the biomechanics of cells in these given conditions, and also what is the biomechanics of extracellular matrix. AFM can do that because if we see here in the simple model extracellular matrix, this is our adhesion points, which are marked here, you know, in this lighter spots. This is the adhesion spots. Then if we change something here from the bottom, then obviously, because focal adhesion points, they are somehow connected with the cytoskeleton. So we do something from the bottom, cell is changing its organization, and then we can feel it using the AFM, what are the changes. And this is somehow known because if we are just thinking about different types of diseases, then we can see that there are many diseases in which mechanical properties changes of mechanical properties are present. Some of them, they are obvious. For example, muscular dystrophy is a disease which by one genetic mutation, there is no connection between the cell membrane and the actin cytoskeleton. That's why muscles are weaker. But cancer, of course, there are multiple genetic changes, so you cannot attribute mechanical properties to one genetic change. But this is like a manifestation of the disease. Fibrosis in our liver, this is also the case when we observe macroscopically what are the changes in mechanical properties of the tissue. There are many techniques using which can be applied to study mechanical properties of single-cell lever, micropipette aspiration, xanthoid enter, fluid shear forces, optical tweezers. The choice depends, of course, on the force and what you would like to study. As I said, we would like to use the AFM. AFM is nice. This is for spectroscopy. It is slow. However, you can place all samples in one dish. So then you are minimizing the conditions of the measurements of the calibration. If it is done everything in the same medium, then the calibration issue somehow is eliminated. And then just you can compare mechanical properties of different samples in relation to reference glass surface. Glass or petri dish surface is very stiff for the AFM because the range of the forces we are applying, maximum like 20 nanonewtons, is not deforming mica, it's not deforming glass surface, and also not deforming petri dish. So everything is related, and here I have found in the internet few years ago, I like this image. Even I don't know who did it, but this is present on this web page. Then you see the AFM tip, which is indenting, which is going inside the cell. And this red lines, this is the actin cytoskeleton, which were fluorescent dye, fluorescently stained with red dye. In our case, we are using green one. And of course, I would like to present some results. We are using the human bladder cells because this is our very beautiful model system. It is measured over the time independently. Who is measuring, then always we see the difference. Moreover, the difference between reference, non-malignant, or I call them normal cells, and the cancerous one is independent also on the fact how they are growing. So you can measure them as a single cells, you can measure them as a cluster of 5 to 10 cells, you can measure them as a monolayer. Always it will be the difference between normal and cancerous cells. But apart of this model system, there are plenty of data showing that indeed cancer cells, they are more deformable. Here there is a comparison for different types of cell lines, which was measured, and the list is not complete. And I would like highlight you only melanoma. Why? Because for the melanoma cells, it's difficult to find normal cells. Usually, we know that melanoma is coming from melanocytes. Melanocytes, this is a primary cell culture, which is difficult to handle. So we are trying to find some reference cells, and then we found the relation that if you are taking cells which are coming from less advanced cancer progression, from the less advanced stages of cancer progression, they are stiffer, they are more rigid than the cells which are taken from advanced stages of cancer progression. So then we can say that indeed cell, and it has been shown also for ovarian cancers, that cells gain more deformable phenotype within the time course of cancer progression. We can attribute mechanical properties to actin cytoskeleton by adding to cells cytochalasin D. Cytochalasin D is a compound which destroyed actin cytoskeleton. So these red nice lines, they will be totally depolymerized. And then this results in a increased deformability, and indeed after treatment of non-malignant cells and cancerous cells, then we see that Young's modulus drops. Decrease of the Young's modulus tells that this is an increase of deformability of cells. Moreover, in the work, there's a work which was done by students which was coming from Ricardo Garcia lab. We found that this is not only the organization of actin cytoskeleton who plays a role in the maintaining mechanical properties of cells, but also amount of actin also will be related to mechanical deformability. And then, of course, measurements on single cells, then we have very clear system. But clinicians, they are just saying, okay, we don't have single cells. We can try to remove cells from the patients and try to say whether the cells is normal or cancerous, but this is not a real case. Could you do something with some tissue? Then we said yes, but in case of tissue, I would like to show you only like two, three images. The interpretation of the data is more complex. Why? Because it's not only actin cytoskeleton who is, or internal structure of the cells who is linked with mechanical properties, but also cells, when they are cultured as a monolayer resembling some model system of the tissue, they produce their own extracellular matrix. This is a comparison between non-malignant bladder cells and the cancerous bladder cells. As you see, these orange fibers, orange structures, this is a fibronectin, the protein on which cells they are sitting. In cancerous cells, there is no, but from the other studies we know that they replace fibronectin to laminin and to collagen. So basically we can see, okay, the structure, collagenous structure is more stiffer than the fibronectin-based one. So also would influence to the mechanical properties within the tissue. Because if we are making measurements of tissue piece in the AFM, we cannot see what we are measuring. We just see, yeah, there's a piece, sorry for that piece of meat you are measuring. Then, indeed, there is the relation between the elasticity of cells, this is a normal and this is a cancerous cells, on the substrate stiffness. This is also in a model system in which polyacrylamide gels were modified with laminin this time. And then you can see that depending on the stiffness, there is a different morphology of the cells, but also there is a different stiffness. The good thing is that after certain threshold value, the mechanical properties somehow are stabilized. However, and another good thing is that independently of the stiffness, if something is cancerous, yeah? Maybe not for all cell types, but for most of the cell types, this is true, so this is a good news. And then this is my last slide, which I would like to show you on tissue measurements. Although we did measurements on cancerous cells, but I would like to show you the results which were submitted by us on multiple sclerosis. Multiple sclerosis is a disease for which nowadays there is no cure. Of course, we can treat patients and then we can get some information, I mean some release of this or some sort of the cure for the patients for the next attack of the disease. We used mice model system. Why? Because in this mice model, we can simply induce, using special tool, special kit, we can induce this type of the disease. And then this is it is possible to evaluate also the clinical score, which is also used for the human patients to determine what is the degree or severeness of the disease. And as you can see, at certain time, up to 10 days, there was no clinical symptoms. Then there is a maximum of the disease. Then there is some sort of the recovery, and this is like a chronic stage of the disease. And tissue, spinal cords, were taken at pre-onset, onset, in the day where there were no symptoms, where symptoms were weak, and the maximum appearance of the disease symptoms, and also in the chronic phase. And then what we did, we measured the elastic properties of white and gray matter. Gray matter is not affected, everything is, done in white matter. And what we see that we have already in the onset phase, when clinical symptoms are very weak, we see the huge increase in the elastic properties. So this means, and our hypothesis is that probably, we don't know what, but something is changing mechanical properties. There are some works which are telling that if we making the stiffening of endothelial cells, then the blood-brain barrier became loose, and it opens some holes for our immunological cells, for lymphocytes, and they allow them to enter the brain. When they enter to brain tissue, they have to move, they have to migrate. Lymphocytes, they migrate on stiffer matrix, not on a softer matrix. So basically, so once they have some conditions to migrate, so basically they can attack our nerves, and destroy them. And this is presenting some sort of the summary image here, in which we see that this inflammatory, it means the attack of this of cells of our immunological system, is mostly visible in the peak phase. And after peak phase, when they really attack our nerves, we see demyelization and axon damages, so we really see damages of our neurons. But we see that something has happened during the onset phase, and also we see white matter change of the elasticity. In fact, it should be the same size of the line, like here, because within the pre-onset, all these stuffs, they are more or less the same. However, I multiply by 10, not by 10, by 3, in fact, just to show that Young's modulus decreases twice. We are not expecting in the tissues stiffness changes like, 1 to 10. Because 1 to 10 would be harmful for us. Also cancers, they will not change the stiffness 1 to 10. They will change slightly, and the slight difference is enough for allowing the disease progression. And this is my summary, so I hope that I somehow convince you that it is possible to use the AFM for biological samples, especially when we are working in a force spectroscopy mode, which is tedious, but it gives really, in my opinion, nice results. This is we. So on your right-hand side, you have the group. And of course, all these measurements we did with the collaborations with biomedical people. I mean, this is we have to gain some biological knowledge from other people. We are thankful for our funds, and I'm obliged also to announce that we got recently EU ETN project within which we employ 15 ESRs and the main aim of the project is to make the standardization protocol for the measurements of mechanical properties of cell tissues and extracellular matrix, aiming the clinical world. And this is rather a message to Park System that although I have 2 AFMs, one is GPK and the other is your system, very old one, still all measurements which I showed here, they are done by this type of the instrument, which is really working nicely. However, I speak it loudly, software for force curve analysis is not very well developed. Thank you.
Thank you very much.